Okay, good morning. Today we're going to continue our discussion of inborn errors of metabolism. Okay, this is a very general sort of principle that many genetic diseases result from defects in the ability of specific enzymes to catalyze their reactions. And the results are compounds upstream of essentially this metabolic block will accumulate to toxic levels, whereas downstream products might be in short supply. And which is more important in any particular disease depends on exactly what those metabolites are and what their roles are in the cell. So we're talking about phenylalanine and phenylalanine hydroxylase earlier. We're going to today talk about the tyrosine pathway, which is downstream, of course, and the diseases that result from defects in that pathway. Okay, remember, our dietary protein is a source of those amino acids for us, and we actually take in through our diet much more than we need to make our own proteins. The rest is degraded and ultimately excreted, and there are problems that occur if those processes will fail. Now, tyrosine is a different example from phenylalanine because it is used metabolically to make important things like melanin and dopamine as well as just the synthesis of our own protein. Okay, so it's like PKU, but not quite, because this is going to be used for various purposes, incorporated into proteins, of course, used for synthesis of melanins, hormones, but the rest is degraded, and that degradation process is important for our health. Okay, so there are diseases that result from lack of tyrosine going into a pathway, that example being albinism, where the idea is that you need your tyrosine to create your melanin pigments. And the opposite, of course, is when the degradation process fails. And that's going to give diseases tyrosinases types 1 and 2 and alkaptonuria. Okay, so this illustrates kind of briefly where those processes are, starting from tyrosine. A block in the ability to synthesize melanin, which is the skin and eye pigments, leads to albinism. And we saw PKU. This pathway doesn't show all of the problems that can occur if the degradative pathway occurs. It's got one of the diseases, alkaptonuria, but there are two others that we will discuss, which is two forms of tyrosinemia. Okay, so the melanin, tyrosine is a precursor. These are pigments found in our eyes and skin. Tyrosinase catalyzes the first step of melanin biosynthesis. And we don't need to know these, obviously, but if we don't have the ability to make melanin, we will have the absence of a substance that is required for the synthesis of those pigments. So here's your classic melanin pathway. Tyrosinase is the first enzyme in that pathway, and that is directly from tyrosine, which is the beginning of the synthesis of both types of melanins, the pheomelanins and the eumelanins. So in the absence of that enzyme, that entire process fails, and you do not get the production of those pigments. Okay, this girl has albinism. You can see that she has little or no pigment in her hair, skin, or eyes. Okay, albinism is a recessive trait, and this particular person is clearly homozygous for it because she's showing the disease. Okay, it's important to realize that there are numerous genes which, if mutated, can produce albinism, not just a defect in tyrosinase. Okay, and the severity of the defect depends on the allele, just like for PKU, where sometimes the protein is somewhat active, but not completely well. And you can't tell what the defective gene is by looking at the phenotype, because many different genes produce the same phenotype. Okay, so in this example, tyrosine is used to create new substances in the body, not degrade them. Okay, so the other diseases which occur as a result of the failure to degrade excess tyrosine, or to do that efficiently, include the tyrosinemias, types 1 and 2, and also alkaptonuria. Okay, these vary greatly in severity according to where the problem is, owing largely to the metabolites that will accumulate in the absence of the enzyme that's involved. Okay, here's one representation of this pathway, and we're going to consider 3 diseases actually that result from deficiencies in enzymes along the same pathway. The first one is tyrosinemia type 2, and that's because the enzyme tyrosine aminotransferase is deficient or less active than it should be. Okay, tyrosinemia type 3 is controversial whether it exists or not, but it turns out that that's an important enzyme in the degradation in ways that we'll see a little bit later. The third one is alkaptonuria, that is deficient in the enzyme that converts homogentic acid to mallyl acetoacetic acid. From there, mallyl acetoacetic acid is converted to fumaryl acetoacetic acid, and then further down for degradation. So the ability to conduct correctly these various steps in this process lead to at least 3 different types of genetic diseases, which differ considerably in their severity, as we'll see. Here's another representation of these pathways, and I'll come back to this in a sec. Okay, so tyrosinemia type 2 is caused by a deficiency in the enzyme TAT, tyrosine aminotransferase. Which of the following compounds will accumulate in these individuals? Phenylalanine, tyrosine, 4-hydroxyphenylpyruvic acid, all of the above. Okay, the answer B for sure, tyrosine will accumulate. So we haven't even managed to get past our first step in this degradation pathway. But it's also possible that these other compounds will accumulate depending on how reversible those reactions can be and the interactions between those particular metabolites. The answer is clearly B, but others can be considered as possibilities. Okay, would a low-protein diet help in the management of tyrosinemia type 2? Yes or no? Yes, we think so, because if the problem is accumulating extra tyrosine, if you keep your protein diet low, or at least your tyrosine intake low, then a low-protein diet will help to control your tyrosine levels. It's like PKU in that respect. Okay, our next disease is alkaptonuria, and this is a really famous genetic disease because it was the disease for which the term inborn errors of metabolism was actually coined. It was the first disease to be specifically connected to a specific enzyme deficiency. Okay, so the enzyme deficiency here is homogentic acid oxidase, which is conveniently abbreviated HGD. And what it does is it takes the homogentic acid and converts it to downstream products for further processing and excretion. So somebody that's missing this homogentic acid oxidase enzyme will accumulate homogentic acid in their tissues, and accumulate, and it turns out a polymer as well. Okay, so this compound or its polymers will accumulate and will be excreted in large measure in the urine, and the polymer itself, which is formed by oxidized products of homogentic acid, eventually accumulate in the tissues to cause a very painful arthritis in people that are afflicted with this disease. The most telling characteristic about alkaptonuria is the metabolic accumulation of homogentic acid, which results in darkly colored urine, which turns black upon standing. So this can be noticed in even very young children, and it's a non-fatal condition, but it becomes more painful later in life as the connective tissue accumulates the polymers that form from the extra. Now, a lot is excreted, but there's plenty left in the body that's not, that causes these deposits, which are very painful. Okay, tyrosinemia type 1 is the worst of these. It's caused by a deficiency in the enzyme Fa, fumaryl acetoacetate hydrolase. This is a very late step in the degradation. It's a very severe form of the disease because you get accumulation of 4 different toxic metabolites which cause liver cancer, liver failure, and the disease is usually fatal except if you can get a liver transplant. All right, what compounds will accumulate in the absence of the Fa enzyme? Please write these on your worksheet. So you need to look at your worksheet and look at the pathway and see what's going to be happening. Okay, so you can see on the handout that you're going to accumulate 4 toxic compounds because the first 2 can be interconverted. It's a reversible reaction easily, and both of the other 2 are formed by spontaneous really metabolism of those 2 other intermediates. So these 4 compounds are really quite toxic and lead to severe liver damage and cancer if allowed to accumulate. Okay, so here's our pathway again, and we are going to look at further steps or other aspects of this pathway. Okay, nitisinone is actually the first successful treatment of tyrosinemia type 1, which seems odd. It's a weed killer. Okay, the weed killer is a therapeutic drug. Nitisinone inhibits the enzyme HPD, and so that inhibits the formation of the homogentic acid, thereby preventing accumulation of it and the formation of the deposits that are causing problems for people. Now, it's a weed killer because if you're a plant, you need homogentic acid to make your plastoquinones, etc., for photosynthesis. We don't need to make plastoquinones, nor could we if we needed them, and so it's not harmful to us particularly, because even though it kills the plant, it's not going to kill the people. Okay, so you need to look at the pathway and suggest how this drug, nitisinone, can be an effective treatment for tyrosinemia type 1. Please answer on your worksheet. Okay, answer. Nitisinone prevents the formation of the toxic compounds because it blocks the pathway upstream of them. So it basically doesn't get rid of them, it just prevents them from forming in the first place because the precursors can't form, and so there won't be any accumulation of these terrible compounds. Now, of course, it has problems of its own in the sense that you're blocking the pathway and you're going to have accumulation of upstream compounds, just like you do for the other metabolic blocks that can occur. Okay, so answer. Patients being treated with nitisinone are told to limit their protein intake and or go on a tyrosine and phenylalanine restricted diet. Okay, so this makes sense because nitisinone, in a sense, creates its own form of tyrosinemia in that compounds upstream of the its block are going to be accumulating. And that's not so great. You don't want to be accumulating too much tyrosine and tyrosine metabolites. And so it makes sense to restrict your intake of tyrosine in order to prevent that from happening. It's not as bad as the real disease. It's a disease in and of itself in a sense, but it's going to prevent the lethal effects and liver failure that results from this form of tyrosinemia. All right. So yes, a low protein or low tyrosine diet will mitigate the problem of accumulation of tyrosine and its metabolites. Okay. Nitisinone is also used as a treatment for alkaptonuria. And on the handout, we see a graph of a study in which some people with the disease were treated with nitisinone to see what the effects were on the accumulation of the homogentic acid in their urine, and also versus a control of untreated people. Okay, so we can look at the graph, and we can see that the y-axis, which is illegible, is the homogentic acid concentration in the urine. The blue at the top is untreated control people, and the duration of the study was 36 months, so it's quite a long study. The important part here is the red line on the graph, where people that began the treatment started off with high levels of homogentic acid in their urine, but within a few months of treatment or of taking this drug, it went down to a normal, really wild-type level. So clearly this drug does definitely decrease the accumulation of homogentic acid in the body, which is a good thing. Okay. So the drug, you can tell from the data, is effective in preventing homogentic acid production, which is what we're trying to do. On the other hand, people that already have the arthritis are not helped by this drug. It prevents it from getting worse in the sense that their homogentic acid levels will go down and remain down, but the deposits that are present are not cleared out by the drug. So it can make it not get worse, but it's not going to get rid of the deposits that are already there. The polymers have already formed. So for newly diagnosed people and younger people that haven't yet gotten the severe arthritis that can result, this is a good thing. For older people, it'll help probably. It won't get any worse, at least.